And the title of our talk tonight is Pollinators, Native Prairies and Conservation by Dr. Susan Waters. She is the founder and senior research ecologist at Quamash Eco Research. Her research focuses on plant pollinator community dynamics in Cascadia prairies. She earned her doctorate at the University of Washington, where her research focused on native exotic plant interactions mediated by pollinators. She currently studies how prairie plant pollinator networks change as sites undergo restoration and as native plant populations rebound. We heard from Dr. Wilderman, from Dave Wilderman and Regina Johnson on our native Washington prairies. And so this is a follow-up presentation to that that's focused on pollinators. So go ahead and thank you for being with us, Dr. Waters.
Thank you so much for having me, and I apologize for arriving in such an unexpected way. It was quite a last 25 minutes. Excuse me. So I will go ahead and share my screen, and why don't you let me know if you can see that?
Yes, we can see that.
Great.
If you could make it the presentation view, that would be great.
Yep, we'll certainly do that. Whoops. Let's go back to the beginning and start with the beginning rather than with the questions. Ta-da. Okay, great. So welcome. I'm going to take a deep breath. So I do indeed study pollinators in native prairies, and I'm particularly interested in how pollinator communities respond to restoration. I'm not by any stretch particularly more knowledgeable than anyone else about Lepidoptera, and certainly am far less knowledgeable than this group. So by all means, contribute ideas if you have them. I have put a couple of slides in here with moments for questions. So hopefully that will allow you to kind of break in the flow. I'm also happy to be interrupted with questions. So I work in the prairies that I guess you have all recently heard about from Dave Wilderman and from Regina. And these are beautiful prairies on the west side, and they're among the rarest ecosystems in North America. And you can see a map in the lower right-hand corner of the screen that shows you kind of the historical extent of those prairies from the central Willamette Valley up through our region and towards Vancouver Island. And they're very beautiful places. What I'm interested in these prairies is the fact that because so many of them are lost, many of them are undergoing restoration. In other words, we're trying to bring back to degraded sites a flourishing and functional native plant community. And that's been very successful in some areas. But what I'm interested in is what happens next. What happens when all those native plants come back? How do all the insects respond to that? So I'm going to give you a little kind of theoretical framework of my talk. It's got a little bit of ecology speak in it, but hopefully that will come across clearly. So I like to think, I think about restoration as a theoretical ecologist does, as informed, intentional manipulation of a plant community, right? You're starting out with a plant community that you don't want, and you're going to do some things, some ecological things that are going to change that plant community into something that you do want. And the things that we usually manipulate in these ecosystems are three things. The first is disturbance. And that's because these prairies are historically disturbed regularly by fire. And I'm sure Dave and Regina probably talked about that. So part of restoration of these prairies is restoring disturbance to these systems. The second aspect of restoration is propagule pressure, and that's just a fancy term for increasing the number of seeds and seedlings of the plants that you want to be there, increasing the number of their propagules. So we do that by seeding and by planting out seedlings. And the third thing is we manipulate competition. We try to get rid of competition from plants that we don't want, like Scotch broom. in order to facilitate the competitive advantages of plants that we do want, like native plants. So we do all these things in restoration, and we hope that they result in changes to the diversity and the abundance of plants in the prairie. And I have a little cartoon here of what I mean by that. So in this cartoon, you can see an unrestored site that consists of some blue shapes and some green shapes that represent native plants and exotic plants. And you can see that this unrestored site is dominated by the blue exotic plant and has a few natives stuck in there. And that's pretty representative of a lot of our unrestored prairie sites. And so we come up to a site like this, we manipulate these things for disturbance and propagular pressure and competition. And we hope to end up with something that looks more like the cartoon in the bottom that says restored, where we have more different kinds of green shapes. So in other words, higher native plant diversity. We've reduced the abundance of those blue exotic plants. And then we have maybe augmented the abundance of some of the rare native plants. And in that case, I'm using that green plus to represent a rare plant, and that's increased in abundance. So this is the idea, right? We do these things to a site and we hope that we end up with something like that bottom cartoon. But what I'm really interested in my research is what happens next, because we've actually been pretty successful in achieving that restored cartoon. But when we get that increase in plant diversity and abundance, it is going to affect other organisms. We have a lot of other organisms out there. As you know, the organisms that WBA loves to love are all immediately going to be affected by plants. And of course, I study pollinators, as exemplified by this beautiful painting that my friend sent me recently of a bee in a morning glory. So I'm particularly interested in pollinators because pollination is a mutualism. So it's a partnership that affects both partners and that can be subtle for both partners. So you have the bee visiting the flower gets the benefit of having its genetic material transferred to another flower of the same kind, right? If all goes well, and the bee or the other pollinator gets the benefit of some nutrition. And because pollination is a mutualism, it's really important for the outcome of any restoration. And that's because the best estimate that we currently have for plants in general on Earth, it's an estimate made by Jeff Ullerton a few years ago and his group, is that something like 85% of land plants depend on pollinators to reproduce. In other words, they're not going to reproduce if a pollinator doesn't show up and do some pollinating in order to result in some seed. And when we zero in on the native Pacific Northwest prairie forbs that I study at my sites, like the ones in these pictures that you see here, we find that this 85% that's sort of a global estimate is pretty true for our local pollinators as well. And so what that means is that the landscapes that we care about, we don't get to have these landscapes unless there's a functioning pollinator community. Everything that you're seeing in this picture that's flowering requires insect pollination. So this is a really important process, and that's one reason why I'm so interested in the feedbacks between restoration of plants, responses of pollinators, and then how that feeds back to affect the plants. As exemplified in this red arrow, so you have plant diversity and abundance changing because of restoration, and that affects the pollinator community. But because the plants depend on the pollinators, that's going to feed back and then affect the plant community in turn. And finally, the plants and pollinators in the community are going to be interacting with each other in some kind of structured way. And that the way that that plant pollinator community is structured has some repercussions in terms of how resilient the whole plant pollinator community is to environmental change. In other words, how well the system can respond to losing a species, for example. When you lose a species, do you kind of get a collapse because everything is interconnected in a way that's highly dependent? Or when you lose a species, do you not get a collapse because it's connected in a resilient way? So these are the questions, the research questions that I'm interested in. Now, this framework that I've put on the page here, this is how I'm going to structure my talk. I'm going to talk about each one of these little boxes. So I'll return to these. I feel like I should have put a questions slide right here. Does anybody have any questions at this moment before I move on? You guys with me?
Yeah, we're with you. I do have a question about pollinators and whether you track them as you look at restoration and whether you see shifts or whether the pollinators are relatively nonspecific.
Yes, I will get far more into that in a few minutes. So maybe that's an excellent question. Maybe hold it and see if I do actually answer it, and if I don't, then we can come back to it if I don't address it to your satisfaction. But let's start with just the plant part to lay the groundwork. So here's the restoration. I'm going to go through this quickly because I think that you've probably seen some of this from Dave and Regina. So here's an example of an unrestored prairie. Lots of non-native flowers in this picture, very undiverse, not a lot going on, right? And we show up and we burn the prairie to restore disturbance. We grow native seeds and we plant out seeds and seedlings in the prairie, as in the upper right-hand corner and the lower left-hand corner. So that's increasing the propagular pressure. And then we might be out there spot spraying individual, say, Scotch broom or tall oat grass to try to diminish the competition from those invasive plants. So there are those actions that I was talking about. And what does it actually look like when we do those actions? How does the plant community respond? We have really visually clear changes in the plant community from doing those actions, much increased native plant diversity. And I find it really difficult to take good pictures of plant diversity. I don't know if any of you has had that experience, but this is among the best I've been able to do. So you can see a lot of different leaf shapes in this picture. You can see a number of different flower colors and architectures. So definitely we do get increased native plant diversity, and we can document that with data. Here's some data from 3 sites. Bald Hill is in Corvallis, Oregon. Smith Prairie is on Whidbey Island, and St. John's Prairie is near Portland. And these are figures showing floral richness, which means the number of different kinds of plant species that are flowering. The red is restored and the blue is unrestored. So you can see that at all these places, when we do these restoration actions, we get more kinds of flowers flowering. So that's very clear. And we also get higher abundance of rare plants, like this beautiful golden paintbrush. So the plant diversity and abundance definitely responds. How does the pollinator diversity and abundance respond? Well, pollinator diversity clearly increases with restoration of floral diversity. And this is an exciting result because it was not a foregone conclusion. I'll back up a little bit and point out these are really highly fragmented ecosystems, and they've been fragmented for a really long time. They've lost many of their plant species. So it has always been a question in my mind, if you build it, will they come, right? If we put back native plants, do we magically get pollinator diversity back? Where do they materialize from? Have they been in refugia? Or are most of the specialists gone and we only get back generalists? These are the kinds of questions that interest me. So it was very exciting to see this relationship. And so this, I'll talk about the graph first. The graph shows floral richness. So that's number of flowering species at a given time of the season on the x-axis, and on the y-axis, pollinator richness, number of different kinds of species. And this is at restored and unrestored sites. You can see the blue is restored and they have lower floral richness and lower pollinator richness, and the red is restored. And so definitely we see a relationship there. And just to dig into some more details, I've included the list over on the left. It's a little bit of a funny list because it combines, it's mostly a list of bee and fly genera with occasionally a random other character thrown in. But that list is there to just sort of exemplify just how much diversity we do see at the level of genus. So this is not even going down to the level of species, but we see a lot of diversity at the genus level in our prairies. And along the bottom here are some pictures. These pictures, as virtually all of the good pictures in my talk, have all not been taken by me, but instead by a fabulous entomologist and photographer, David Kappert. So I want to acknowledge him here. So on the lower left-hand side, I've included a bunch of mug shots of a group of pollinators called the osmia, or, you know, they're often called mason bees. We have really high osmia diversity across our sites. It's been really cool to see what kind of diversity we have. And if you look carefully at those little blue bee faces, you can really see there's some variety there. It's really exciting. And the picture in the lower right-hand corner is just zooming in on one of them and showing you how we document. A lot of our pollinators that we capture can't necessarily be identified to species because some of the keys are incomplete. And so we have to document or are changing. And so we have to document our morphospecies, and because they may change later on as the taxonomists make their decisions. So pollinator diversity is definitely increasing as we restore floral diversity, and that's really exciting. So who's actually coming back? Our west side Cascadia prairies are very dominated by bees and by flies, as exemplified in this figure. This is 2 different years. This particular figure is showing you the same 3 sites that I talked to you about before: Bald Hill, Smith Prairie, and St. John's. And the y-axis is the proportion of the observations that belong to the different groups of insects. And they've been lumped, the insects have been lumped into guilds that are loose associations that are sort of ecologically similar in terms of their pollination effectiveness. And red is bees and green is flies. And you can see that most of those, a really high proportion is red and green. You can also see how few Lepidoptera we detect. That's a tiny slice in there. Now, these data come from observations, timed observations at floral patches. So we sit at a patch of flowers. The patch of flowers has to be bigger than a certain, bottom number so that we know that we're at an attractive enough patch to be drawing insects. And we do timed 15-minute observations, 2 of them at every single flowering species at a site. So that's where these data come from. It's been an ongoing topic of conversation amongst my field techs and how few Lepidoptera seem to show up and visit flowers while we're watching. So that's an interesting conversation to continue perhaps with you guys later on in the talk. If we look at 3 very, very, very gross categories of bees: bumblebees, which are native eusocial bees; honeybees, which are non-native eusocial bees; and solitary bees, which is a giant group. When we break down into these categories, again, red is restored, blue is unrestored. Pretty much no matter where we look, we find there are more bees of all kinds at restored sites than unrestored sites. In addition to just wanting to know about who's out there in terms of the pollinators, we also want to know which of our plants are especially vulnerable and which of our rare plants are especially pollinator dependent. And I guess I do mean vulnerable in terms of needing pollinator services in order to make seed. And I'm actually gonna just skip, I'm not gonna delve deeply into these figures, but just briefly to say that I do work with two species that are federally listed, Castanea levisecta, which may become delisted soon, and Erigeron decumbens, that's golden paintbrush and Willamette daisy. Both of them are pollinator dependent. Golden paintbrush is super pollinator dependent. You can see in the lower left-hand corner that if you give bees access, which is that category called open, versus if you exclude bees from the flower, that category called self, so self-pollination, the self-pollinated plants make 0 seeds. So it's very dependent. And we do see competition from other plants on the stigma. So in the lower right-hand corner is a scanning electron micrograph taken by Savannah Richard. And that's the stigma, the receptive female part of a Willamette daisy flower. The nearest kind of, what would you call that? Like a spiky soccer ball structure on there is pollen from the same species. So that's pollen that is likely to pollinate that plant and has probably been transferred by a visiting pollinator. The other two are pollen that came from a different species. And so they are clogging the stigma and taking up space that could otherwise be taken up by pollen of the correct species. So that's a sort of pollen competition at the stigma. Talking about Willamette daisy is my opportunity to show you some great photographs of David's. The next few slides I'm going to show you are just a series of shots showing the incredible pollinator diversity that is supported just by this single endangered aster species. It's the diversity is remarkable. So these pictures are all showing halictid bees. For those of you who are keeping score, these are Lasioglossum and Halictus foraging on Willamette daisy. But we also see cuckoo bees, like the Coleoxys in the upper left-hand corner. We see Nomada, like the cuckoo bee in the lower right-hand corner, the red one. Just everything you can imagine shows up on this flower. In this slide, we have in the upper left-hand corner a longhorn bee. There's a Melisodes. I'm going clockwise. So the longhorn is in the upper left, then a Melisodes with that marvelous thick pack of pollen on her hind leg. A leafcutter bee and a small carpenter bee. So this is a lot of bee diversity, and that's before we even get anywhere near the flies. And there's a ton of fly diversity, and I'm only showing you a fraction of it. The upper left-hand corner is probably a less common pollinator. The other three are syrphid flies or flower flies. And then occasionally we'll see a butterfly or a moth. They do show up. So that's kind of an overview of the sort of the way that we're approaching our work in terms of thinking about restoration influencing plant communities, in turn influencing pollinator diversity. And we do see pollinator diversity springing back, and a lot of pollinator diversity on specific plants, including endangered plants. And we do see that the endangered plants in turn themselves, some of them are highly pollinator dependent. So let me pause there and ask if there are questions.
Yeah, we do have a couple of questions, Susan. First of all, your photograph of plant diversity I thought was absolutely marvelous. I thought you captured a lot of different species. And there's a question, somebody else felt the same way. So David Jennings is asking, how difficult is it to get your seed sources for the native plants? Because your, your results are just astonishing.
Oh, credit is not due to me for any of that actual restoration work. Most of that restoration, that particular picture was actually taken on Johnson Prairie, which is part of Joint Base Lewis McChord. The Center for Natural Lands Management has been deeply involved in that restoration, and they operate a couple of different sites that are native seed farms. So they collect seed from extant populations, and they then grow it out in native seed farms, and to amplify it. And then they either plant the seed itself or grow the seed out of seedlings and plant those seedlings out. And that's in combination with the strategies of returning fire to the system. And they still do use herbicide, mostly in the form at this point of spot spraying individual plant competitors.
Okay, that's great. Another question is, are your three sites, the Bald Hill, Smith, and St. John's, open to the public?
Oh, great question. Smith Prairie is open to the public and loves to have visitors. They are, the ownership there is Pacific Rim Institute. It's up on Whidbey Island, just south of Coopville. St. John's Prairie is a restored landfill in the city of Portland. It is not currently open to the public, but it will be, is the plan. And Bald Hill is open to the public. Yep.
And where's Bald Hill?
Bald Hill is in Corvallis, Oregon.
Corvallis, that's right, okay. Okay, and another question about the species of the aster curtis.
It's Erigeron decumbens, yeah.
Okay, great. Go ahead and continue, Susan, thank you.
Okay, excellent. Good question. Okay, so I've kind of given you an overview of plant diversity and pollinator diversity. Now I'm going to move on to talking about whole communities of plants and whole communities of pollinators and how we're finding that they're structured and interacting. So I use an approach called network theory to study plant-pollinator interactions. And this is an example network. Don't look at it too closely because it comes from way back when I didn't, my insects weren't identified as species yet. But the gist of how you look at this picture, I'm going to show you a whole series of pictures like this. So explain how to look at it. Along the bottom, you'll see that there are 1, 2, 3, 4, 5, and a little teeny sliver of a 6th black rectangle. Okay, each of those rectangles represents a plant species. Up above, and that's why there's a red flower next to it, that's a castellea. Okay, up above there, the bombus is representing the insect species. So that whole series of black rectangles, those are all the insects. Every time a line connects one of the insects with one of the plants, that means we witnessed that interaction. Okay, so we saw that pollinator or flower visitor land on that plant's flower. The thickness of the line is proportional to how often we witnessed that interaction. So the cool thing about these images is that you can learn a lot right away. For example, you can see that there's a really big, giant black rectangle on the bottom, and you may not be able to read what it says underneath, but it says hyperad, and that is a nickname for Hypochorus radicata, which is a noxious weed that everyone loves to hate. Turns out it's supporting a very wide variety of pollinators in this particular site, right? This is a very unrestored site. So guess what? The pollinators don't have anything else to eat. So it is actually quite a central plant at this moment for these pollinators at this site. So you can also, you can see, the other thing you can see at a glance at this and these networks, as you can see, there's a bunch of insects there that are only connected to one plant, right? So that means that those are insects that we automatically would have some curiosity about, like, gosh, maybe we should do some more observations and see, are they totally dependent on this plant, or do they really visit more things and we haven't detected that yet? So I really love this particular approach using networks. It's a community-based approach. You can visualize the patterns of interactions. You can also do some fancy stuff with algorithms that analyze the potential stability and resilience of these networks. And of course, you can view shared pollinators, right? You can see which plants are sharing which pollinators. Now, the picture that I'm showing on the right here is the same kind of network, but viewed in a slightly different way. In this particular way of looking at it, the insects are along the bottom here, where you see the bee icon, and then the plants are along the side. And every time a plant interacts with a pollinator, that square is colored. And the more they interact, the darker the square. Just a different way of looking at the same thing. And it makes it easier to see different kinds of things. But either way, these are ways of looking at communities that I find valuable. So how do we actually get these data? I'm going to show you data from the South Sound that comes from 6 sites that span 0 to about 12 years of restoration and a reference site. It's a little bit difficult to say. We're in the process of using some spatial data records of restoration actions to be able to really explicate, like, how much restoration actually happened in the exact area that we're including in the study. So that's why I say 0 to about 12. So to get these data, we visit each site every 2 to 3 weeks. We assess the floral abundance of everything that's in flower, like the quadrats you see at the bottom. You can take a look at those quadrats and imagine counting flowers all day long. Takes a little bit of work. And then we perform 30-minute observations of pollinator visits at every single forb species, that small herbaceous flowering plant, everything that's in flower. We find patches and we watch what's coming. And everything that visits that plant, we either visually identify it or we collect it. Now, we have a policy of never collecting Lepidoptera because we work in sites that have rare Lepidoptera, and none of us is an expert on Lepidoptera. So we just write down what color they are and hope that we can identify them visually. But since we rarely get them, that's usually not an issue. The other thing that we never collect is queen bumblebees. We exclude queen bumblebees. So those we identify visually as well. But everything else is collected and identified to the lowest possible taxonomic level by people who are more expert than we are, so real taxonomists. And so we have 4 years of data collection at these 6 sites. And here's a sample of the results. This is a site called Glacial Heritage. I'm sure many of you are familiar with this site. It's in the South Sound. Again, plants are across the bottom. Pollinators are across the top. I've color-coded it by sort of rough pollinator guilds. Red is bees, blue is wasps, tan is flies, green is leps, and gray is all others. So that would be, beetles and other stuff. And you can see at a glance, boy, this is a bee and fly system. Very bee and fly dominated. So, and it's complex. At this point, I'll pause and say we have about 8,000 specimens now, which is the largest collection in existence, as far as we know, for West Side Cascadia prairies. So working to expand that as well. Okay, so now that we have these cool looking structures, how can we use them? Like, what's the point? How can we use the information on these community structures in restoration and conservation? So I'm going to give you 2 examples of how we think they can be used. And here's example number 1. Example number 1 relates to Taylor's checker spot butterfly. So Taylor's checker spot, as I'm sure many of you know, is federally endangered, and its larvae are specialists, and they must feed on specific host plants. In the prairies that I work in, there are only 3 main host plants that they're laying on. 2 of them are insect pollinated. 1 of them is wind pollinated. I'm not going to talk about the wind pollinated one, but I am going to talk about the other 2. One is harsh paintbrush or Castanea hispida. The other one is golden paintbrush or Castanea levosecta, which itself is federally listed. Now, lots and lots of money and time and effort and angst is spent to establish these host plants at sites in order to reintroduce this butterfly. However, both of these host plants are virtually 100% pollinator dependent for reproduction. And when we observe them, it's hard to detect visitation. Pollinator visitation, especially to Castanea hispida, is very low and very specialized. We see Bombus californicus and the very occasional Bombus vosmosensky, but basically entirely one species of bumblebee. So what's interesting about this is, gosh, we really want to have these host plants in order to support this butterfly, but guess what? The host plants themselves require something too. So we can ask this question: what other plants? Okay, well, let's go backwards here. We start with the butterfly. So I'm at the butterfly and going backwards against the flow of the arrows. So starting with the butterfly needs the host plant. The host plant requires the pollinator, but the pollinator in turn visits a lot of different plants, right? Pollinators live longer than the flowering of any one plant typically. So a pollinator is visiting a lot of plants over the season. So we can ask, in addition to the plant that we care about, like the host plant for the butterfly, what other plants do we have to have in our system in order to support the pollinators that support the butterfly host plants that support the butterflies? I hope I made that clear. And we can use our networks to answer this question. So here's one example. Again, plants across the bottom, pollinators across the top. This is golden paintbrush. I've stuck it in there in front of its little tiny black rectangle. You can see that at this particular site, in this particular year, we only detected 2 different kinds of visitors to golden paintbrush. Let's follow one of them. One visitor was Bombus californicus. This Bombus californicus, however, it's a little hard to see in this picture, but Bombus, this bee itself, also at this site in this year, was only ever detected visiting one other plant besides golden paintbrush. So what was that plant? It was a plant that blooms so much later that it doesn't even overlap in flowering time with golden paintbrush. So this is a really interesting thing to see because it raises new hypotheses that we can test. And the conservation implication is that plant species that bloom at totally other times than the butterfly host plants that we're trying to care for, like this purple flower, Prunella vulgaris, may be important to the pollinators that we have to support the populations of to carry over to next year to pollinate the butterfly host plants. So we think this is an interesting use of these networks. Here's another example. Different site, different year. This is golden paintbrush again. You can see 3 different lines coming out of that golden paintbrush. One of them is a fly that probably does next to nothing. I'm going to follow this very, very, very common bumblebee, Bombus vosmosensky, visited golden paintbrush. And at this site, despite the fact that this bumblebee is ubiquitous, we only ever detected it visiting one other thing at this site. What was it? It was a weed. Same weed I mentioned earlier, Hypochorus radicata. So again, this also has conservation implications. As we are getting rid of weeds or non-native plants at a site, we have to remember the fact that there are other species besides the plants, and that some of the species that we may really care about and that may have really important functional roles in this system, like pollinators, may be depending on them. So the implication is replace them with something. If you're going to be eliminating this flower, put some other floral resources in place to support the pollinator. I'll pause again for more questions.
Okay, great. Thank you, Susan.
I'm just going to grab my phone. I usually start the timer at the beginning, but I was so discombobulated by arriving late that I didn't do that. So I'm just hold on 1 second. It's right up here.
Paulette, may I ask you a question?
Great. Yes.
May I?
Yes.
Yeah.
Okay. First of all, Susan, bravo. Thanks for mentioning that very last point, which is extremely important and too little mention. I was going to bring up Hypochirus radicata if you didn't, but it's clearly the most important butterfly plant in Western Washington in the latter part of the season. And if we eradicate that in restoration, which we're all behind, then of course we may be having net loss. Second thing is that's not the only one. There are other non-natives, as you mentioned. The prunella is one. It's very important for many species. And of course, as we know, the Castilleias are not going to do it by themselves for the checker spots, which are utterly dependent at most sites on the non-native plantain.
Yes.
That's a big problem now because, as Paul Severance has found, I just reviewed another paper on this, than his first one, there is now a Ascomycetes pathogen that's come in on the plantain and is knocking out a great deal of the plantain. So the checker spot might be
that pathogen that shows up in the wintertime. Yeah.
Right. So the checker spot might be in big trouble regardless, except on the sites where the little veronicas will grow. But I wanted to ask you also about the question of the few records of LEPs that you have in your studies.
Yeah.
With the butterflies, it's clear that if you're not able to identify them on the site, you're going to lose a certain portion of the diversity there. But I suppose even if you only have gross IDs, you're still going to capture the basic data of a LEP visit. But the other question is this: are you doing any nocturnal observations? Because if not, you're missing 90% of the LEP diversity.
No, you're totally right. We're not doing, we're definitely not doing nocturnal observations. And that is undoubtedly feeding into a lower detection rate. No question. Yeah.
Well, thank you. Wonderful talk.
Oh, thanks. Great.
There's comment from Regina Johnson that prunella is native.
Yeah, Paulette, I wanted to make another comment too, as someone whose entire professional life revolves around weed control. There's no chance we are ever going to eradicate the Hypochirus. We don't even try.
I was going to say that.
No, but we can eradicate
it from important carpet. We just don't even try. It's a solid carpet.
No, you're right, and that makes it it's in a way making that point is from one perspective moot, but it's an example that comes up with other species too.
Right. For other reasons.
I just think it's a larger issue, that the idea that we, that it's important to expand our thinking to think about the mutualists who depend on whatever's there now, even if it's non-native, while we are trying to restore the native. There's got to be a way of using heterogeneity and using sort of a stepwise process that can help facilitate supporting the pollinator and butterfly populations that we have, while trying to gently push that plant community back to where we would like to have it.
Right. Yeah, that's a good point.
May I say that we're not going to eradicate Hypochirus radicata for sure, but we can eradicate it and have eradicated it from certain very important sites where certain uncommon species are dependent upon it. And that was the case with tansy ragwort at Cascade Head in Oregon, when it was eradicated, when the Oregon silver spot butterfly was dependent upon it.
Yeah.
So there's another question here from Michael O'Loughlin, and he's asking, what is is your insect data going to be uploaded to GBIF? And I don't know what GBIF is, but
GBIF, yes. We do intend to upload it to GBIF, but there's quite a process to go through before that. So we just it takes a heck of a long time to process our specimens because they go to a fly taxonomist and a bee taxonomist, and then they come back together and have to be reunited, morphospecies unified across years. So we actually just got back our data from this past summer. So it's a slow process, but yes, we hope to do that. That's a way to kind of contribute diversity data to the wider world. So I think I should go on.
Sure, go ahead.
I just wanted to say that Prunella vulgaris comes in a native race and a non-native race. So it's easy to think it's non-native or easy to think it's native, but our native race is Prunella vulgaris, lanceolata, and the non-native Prunella vulgaris vulgaris.
Ah, okay. All right, that's helpful. I wanted to go on and give you a second example of how we think this, this kind of plant pollinator network information can be used in restoration and conservation. So again, here's an example of a network. We can use this in a very simple-minded, not very fancy way to ask a really straightforward question, which is, are we restoring plants that support diverse pollinators? And the reason why we would want to ask this question is not because we won't eventually restore diverse plants that, plants that restore diverse pollinators, but because restoration takes a long time and is iterative. And we're not necessarily planting in the same species every year at every site, right? And so sometimes plant lists can be a little bit opportunistic. Oh, we have some seed of this, so we'll focus on that, or we won't focus on that because we don't have seed. We're suggesting, our idea is, if we know that a certain set of plants are really generalist and can support a wide community of pollinators, planting those seeds early in a 15-year arc of restoration might help establish those pollinator populations so that they're in place by the time later on more specialized plants that require particular pollinators are brought into the scene. So we can do this really easily with our networks. In reality, we do it using an actual algorithm. On the screen, all I've done is put a circle around black rectangles that have lots of lines coming out of them. So you can see there's a lot of plants that are supporting a lot of pollinators. And when we do this, we come up with some of the total usual suspects, right? Camas, which we have at most sites that are prairies anyway, so we're not even bothering to restore it in most cases. Eriophyllum, which is often already there. Plectritis, which is already being planted everywhere. But we also come up with some less expected characters. So for example, we found that Micranthes integrifolia, which is on the bottom there, and Potentilla gracilis were both supporting groups of pollinators that were not so well supported by other plants, or were supporting a very diverse group. So we suggest that including these plants early in the restoration process could potentially help support plants that are planted in later. So that's a second pragmatic use of networks. Paulette, could you tell me, should I stop at 8 or should I just go? I think I'm going to go about 10 minutes over if I don't kind of.
You just go ahead and go, and if people need to drop out, they can drop out. Yes,
Okay.
please. And then they'll probably discussion after that.
I love having the questions, but they stretch it out though. Okay, so now let's turn our attention to what about invasive plants, because some invasive forbs are obviously also visited by diverse insects, like the 3 that are on this page. So how important are these plants to native pollinators? Well, we can do some simulations with our networks. And what we can do is simulate what people are already trying to do oftentimes at some of these restoration sites, which is get rid of specific plant species, right? We can pretend we got rid of them and see what we think would happen based on how we think the community structure looks. And so these simulations use the plant pollinator networks, and they remove every single forb, every single plant one at a time, and then they predict how will the resulting pollinator diversity look. What I'm going to do is I'm going to show you what happens. I'm going to organize it so that it pulls out just the invasive forbs first, and then we'll stop and look at it. So that the image is going to be on the x-axis is going to be the plant species that are extirpated, the proportion of those, and on the y-axis is going to be the proportion of pollinators extirpated. I'm going to start at Johnson Prairie, which is a pretty diverse place. We have 7 major invasive species there that I've listed. And when we look at the first 7 dots in this extinction simulation, and what we see is an estimate of approximately 10% loss of the pollinators when we get rid of those, if we simulate getting rid of those 7 plants. So 10% that's pretty, that's potentially significant. Let's look at a site that is actually a more degraded site, a less diverse site to start with. If we look at Cavness Ranch, we only have to get rid of 4 invasive plants because it's a much less diverse site, but we get rid of those 4 plants and we end up with about an 18% pollinator loss. So this is really interesting. Now, anybody who is familiar with watching bees might already be thinking of a potential caveat to this. This is just a simulation, right? This isn't real life. But first, let's think about the conservation implications. So on the face of it, what this looks like is the same old message. It's important to provide alternative native flowers when fighting weeds. And I do think this is a message that my work brings me back to over and over and over again. Now, there is a caveat though. Okay, it's first, I forgot this slide. It is, of course, it's not that we don't want to get rid of these invasive plants, but we need to pay attention to how we do it. But there's an assumption here, and the assumption is the pollinators aren't going to just go visit another flower. And that's kind of a not very good assumption. Most pollinators are pretty flexible and pretty generalist, and they will often switch hosts. They're way more generalist than at least I believe they're way more generalist than most Lepidoptera are. We do have a pilot study that has been done recently by a student who worked with me. And the pilot study was looking to see how does pollinator behavior rewire these networks when you take away an invasive plant. And we did it with Hypochorus. And we did a before and after study. We had plots. We did the observations to get the network data for that plot. And so in this network, you can see we have 1, 2, 3, 4, 5 flowering species on the bottom. And then we went through with scissors and we cut off the flower heads of all the Hypochorus in the plot. And you can just imagine how long that took. And then we waited 2 hours and we looked at the network again. And it's fascinating. It's totally rewired. And we saw an increase in the connectance of the network. We saw that the diversity of pollinators increased for 2 native forb species. So this is really cool and interesting, suggestive work. It's totally just a pilot, but we're hoping to be able to follow up on it because it would be a great thing to know and very relevant to, again, maybe Hypochorus. We're not at any risk of getting rid of Hypochorus, but again, applicable in general to how systems might respond when we're getting rid of invasive plants. I'm going to skip this question slide and come back to it at the end because we're nearing the end here. So back to my little model here for the overview of the talk: restoration actions, generating changes in plant diversity and abundance, interacting with pollinators to form a community structure. And finally, the last thing on the list here is about resilience, which is really ultimately my interest and is also the hardest thing to really study well. But I do have a few things to say about it. I'm going to show you in sequence the 6 sites that I've been following. So here's the plant pollinator network for a site with 0 years of restoration. 2 years, 5 years, 8 years, 11 years, reference. I think this is really cool. You can totally see complexity coming back to this system. And it's not just, but the crucial thing here is it's not just that more players are coming back. It's also that the players are interacting differently. In other words, when we analyze the structures of these networks, we see changes. The structure of the networks is potentially correlated to characteristics that are relevant for resilience. And I don't have time to dig into profound explanations of these network metrics, but there are 2 of them that I'm particularly interested in. And so I'm just going to sort of, in an abbreviated way, say that these 2 things, they're both written on the x-axes of these graphs that I have here. One is modularity and the other is nestedness. These are characteristics of the ways that interactions are organized between plants and pollinators. And the degree of modularity or the degree of nestedness is thought to be related to how a system responds when responds to species loss. In other words, related to resilience. Our hypotheses, which are drawn on this graph, is that as restoration increases from unrestored to late stage restoration, modularity will go up. We predict that nestedness will go down over the same thing. And again, I know these are just random words, but just trying to give you the sense that there are some metrics that we can measure in these networks and that potentially are related to resilience. And we have actually measured these metrics for 2 of our 4 years. And we find, generally speaking, it's possible that our hypothetical relationships are going to prove to be correct, but stay tuned. So that was a lot of hand waving, but I hope you will stay tuned. Okay. So I'm going to close. I'll just say some of our future directions. We have sites across kind of the range of the latitudinal range of our Cascadia prairies now. We have 12 sites in Washington and Oregon. And so we're hoping to be able to look at a latitudinal gradient of how plant pollinator networks are organized. We're also going to look at how floral and insect traits influence who matches with who in these networks. We're also interested in looking at what happens when the invading Bombus impatiens, eastern bumblebee, disrupts our networks. And of course, we want to continue to use networks in conservation of endangered plants, and that should say, and insects. And with that, I would like to thank my wonderful funders and wonderful, wonderful helpers. So thanks to the U.S. Fish and Wildlife Service, the Department of Defense, the U.S. Army Corps of Engineers, and Jim and Bertha Falconer, and to the incredible people who have done this work in the field with me, as well as to those who have done all the ID work. And I will take questions.
Thank you very much, Susan. This is a fantastic presentation about the complexity of these projects that we undertake. There is one question about your pilot study where you showed the networks being rewired. Did you find that any of the pollinator species did not stay there, that they were actually removed from the system by removing that one invasive species?
That's a great question.
Thank you, David Jennings.
That is a really, yeah, thanks, David. I don't remember. It was like I said, it was a pilot. And so it was just a couple of sites that we did it at. And the work was really sort of being led by Amber Haley, was the student, Amber Haley Kinnaird.
Right, that was, for me, it was more, we're coming to appreciate the native pollinator community is very understudied itself. That the
Oh,
focus
yeah.
has been on the plants, and now, as you were alluding to the diversity of the species on those sites, it just seems like, again, that's going to be a more another place to do more research about all the different species. And
Absolutely. You know what? I just realized I can actually show you, since you're sharing my screen, I can show, this is my website. Quamish Eco Research is the name of my tiny LLC. And if this will load, I don't know if this will load. I'm outside using Anne's Wi-Fi on her porch, which is very kind. It's not loading. If you're able to later, visit my website, and there's some fantastic photos, none of which I can claim responsibility for, all taken by David Kappert, but amazing diversity. And not only does he have photos of, oh, here's some bees on flowers, whole series. Yeah, you can peruse at your leisure. But he also has, you can follow this, says click here for exemplars of pinned specimens. He's been doing some of the focus stacking photography as well. And so he has a whole other page showing some of the diversity of our specimens. And yeah, by all means, take a look. We love to share.
Okay, so everyone is free to unmute at this point, but Bob Pyle has a couple of questions about the definitions of modularity and nestedness, as well as
yeah.
So
I knew that would come up. It's always a part of my talk, and I wanted to at least mention it, but it's so hand wavy to not explain it. And I thought I left an extra slide. Why is this not letting me scroll? Come on. There we go. Okay. For those of you who really want to dig into this, here's the modularity nestedness explanation. Okay, it's going to take a minute. I won't be offended if you guys decide to sign off while I'm talking about this. But okay, let's start in the upper right-hand corner of the screen, where there's a more familiar looking network, right? So VWXYZ, those are the plants. A, B, C, D, E are the pollinators. Everybody can see that. Okay, now we're going from that format of looking at a network to the grid format of looking at a network. It's still a network, but in this case, VWXYZ are the plants along the side. Whoops, what happened? What happened?
it says, oh, there you go.
There we go. Weird. VWXYZ are the plants, A, B, C, D, E are the pollinators, okay? So in nest, so what is happening? Oh, I'm accidentally swiping and it's going black. Okay, sorry. So these two ignoring the red circle for a minute, you've got two side-by-side networks. They both contain the same players, right? They both have VW, X, Y, and Z, and they both have A, B, C, D, and E, right? The left-hand one, it looks kind of random and disorganized. The right-hand one is very organized. This right-hand network is called a nested network. And the characteristic of a nested network is that the ones at the core, that like plant A associates with every single insect, and pollinator A associates with every single plant, and plant V associates with every single pollinator. What it means is that the more specialized plants like these down here, all their partners are a subset of the partners of the most connected characters in the network. Give me a thumbs up if that makes sense, and if not, I'll try to rephrase it. Okay, I've got at least one or two.
yes.
Okay, this structure here is a hallmark of super generalists. So A right here, A is a super generalist. It's associating with everyone. V is a super generalist. Well, guess who our super generalists often are? They're often our invasive species. So these highly, highly nested networks are commonly associated with invasiveness, and they connote more competition among the players. Like there's going to be a whole lot of competition. You know, Z is going to be competing against Y and X and W and V for A's attention, right? So there's a lot more competition here. The competition is not relaxed in any way, so it's potentially more fragile. Consequently, I've put a big red circle around the left network, which is less nested. So we view that as being the more desirable characteristic, or at least probably the more reflective of a healthy and resilient system. The counter argument to that is this right-hand one, you can lose some species here really easily without sacrificing the structure, right? You can lose Z, it's not going to matter. You know, I mean, of course, losing Z is going to matter to Z, but it's not going to matter to other characters in the system. But the flip side of that is if you lose A or V, everything collapses, right? So a nested system is highly stable, but stable in the wrong way. Modularity, so down here, I have two different networks. The left one is non-modular. The right one is modular. Modular just means that you have subsets of species that interact a lot with each other and then maybe are loosely connected. And you can really easily imagine that being a phenological thing, right? You could have a group of pollinators that's interacting with camas and buttercup because they're all early season. And then maybe you have a pollinator that's long enough lived that it's later interacting with a bunch of things in the late season. So those are like early season module, late season module connected by a long-lived pollinator. That's a modular network. Modularity is associated with resilience because you can lose a module without compromising interactions in other modules. It also reflects specialization and reduces competitive interactions. So lower nestedness and higher modularity are the characteristics that we believe are associated with higher resilience. That was long-winded, but I hope it made sense.
Yeah, that makes a lot of sense. Anybody else have any questions? Please feel free to unmute. It's great explanation, I agree. So everybody's free to just interact.
Susan, could you also define what a reference slide is when you said reference? What did that refer to?
Oh, when I said I had 6 sites and one of them was a reference site, it's a term that probably I shouldn't use, but basically my sites are along a gradient of restoration, and one of them has not ever been degraded to the same degree as all the rest of them. So Johnson Prairie, we considered as loosely as a reference point.
Where is that?
It's on JBLM.
On what?
Joint Base Lewis McCord.
Oh, right. So it's like a control almost.
Yeah, it's a comparison. Yeah.
Thank you.
It's the best of what's left.
Yeah, exactly. And it has had some restoration. So it's being burned, but it hasn't had nearly as much seeding needed in order to maintain its floral diversity that already was pretty florally diverse.
I have a question about the differences between your sites. So I noticed that when you were showing the 3 sites of Bald Hill, Smith, and St. John's, that I think it was the St. John's site had a lot more solitary bees than other types of bees. And I was just wondering about how different they are. And I mean, I know you're looking at a, kind of a latitudinal gradient there, but I'm just kind of curious if you could comment on that.
You mean how different the sites are from each other?
Mm-hmm.
They're so different. I mean, this is a real Achilles heel of this kind of study, is that, I mean, I feel like I'm not going to live long enough to get decent data because, insect populations go up and down and all over the place, and sites are different from each other. But they do differ. I was very surprised, actually, to see how much more abundant and diverse the solitary bees were at St. John's. Why? I don't know. I can't answer that question. It is a site it's near water, it's possible that that has an influence.
It's
I
right in
don't-
the industrial part of Portland. Could it have
Right.
anything to do with the popularity of Mason bee houses that so many people put up?
No, because these are not blue mason bees.
Yeah.
You know, we're talking about a whole variety of, you know, a variety of halictids, a variety of andrenids, you know, osmias also. But yeah, no, I don't have an explanation for that. It's great. It was great. It was totally unexpected and exciting to see that they were so successful in bringing these populations back in.
David, did you want to ask your question?
Sure. I just wanted to give other people a chance if they had, but not hearing anybody trying. So early on, you were talking about the golden paintbrush and the California bumblebee.
Yes.
And later on in the season, it was using that blue plant.
Yes.
So,
Prunella vulgaris, the one that's both native and exotic, depending on its
got it.
variety.
Oh, thank you. So what struck me was it had to have been feeding somewhere not on your site in the interim period, right? Because it's a generals that has to have a floral source continuously through the season. So
Well, yeah, yes and no. I mean, we're limited. It would be great if I could say, you know, the extent of our coverage of data is so good that, you know, we're able to detect everything across the whole season. But in actuality, we're going at 4 seasonal time points. So when I say, you know, we only detected it visiting 2 things, that's a little melodramatic, because in actual fact, it's perfectly possible that it had visited some other things at the site. And I'm using that to make the point that those later season flowers can be very important. But if I'm honest, I have to also say it's perfectly possible that we didn't, you know, that it is foraging on site in between and we didn't detect it. Having said that, it is also possible that, especially with the bumblebees, that they are doing some foraging off site. And that,
right.
you know, the likelihood of that is really different from site to site, depending what's nearby, I think.
And then a comment about the field data gathering technique. We had a really nice talk from the Spokane chapter on bees and pollinators on their campus, and they set up video cameras and watched, and
Yeah.
they could observe it over time. And I was just wondering, for example, was there anything that maybe the butterflies were more susceptible to being adverse to coming to where a big mammal was sitting on the ground that the bees were, or something like that, where you would not have that observer bias as clearly with that, and you could also look at it over time.
Yeah, and that is that's an excellent point. We tend to get complacent about our own impact after we've been out there for days and days and days. We forget that individual insects are not necessarily as immune to our presence as we'd like to think.
Also, David, the butterflies are only going to be active in the sunshine, and that's not the case with the bees. And the other thing is that there aren't that many species of butterflies there anyway.
Yeah, we do limit our observations. We only observe when it's warmer than 65 or warmer and cooler than 85. So between 65 and 85 degrees. We have a humidity envelope and a wind envelope also. So I think we limit it to a relatively similar, a set of circumstances that I would expect would be pretty similar for butterflies.
It also might be a way to capture some of the nocturnal visits.
Mm-hmm.
You know,
So
with
we
an
have
infrared, they've really gotten some nice cameras that don't need much light.
Yeah, we have used in the past some small cameras. We don't have so that the challenges with using cameras are 2. First, to have enough of them to watch every single forb species in the place at every site for enough time. And then the equal or larger challenge is to watch all that video. And it's the benefit, as you point out, is capturing data in ways and at times that we can't necessarily capture otherwise. The downside is that we can't take the bees to our taxonomic experts afterwards, and you really can't identify them visually for the most part. So it's
Right.
a balancing act. We do have some small cameras, and we have been toying with the idea of using those in the field and this year in a creative way. If we can just figure out how to, it takes a long time to watch. Every time you put your camera out for 2 hours, then you have to watch 2 hours of footage. So
I like the speaker's comment of something along the lines of, that's why it's nice to have students. So you
yeah,
give them credit for sitting through 30-minute blocks of watching flowers.
totally true. Oops.
There's
Anyway,
a comment.
thank you so much. This is really good.
There's a comment from Katie Buckley. Thank you, Katie. Please feel free to chime in, suggesting that higher solitary bee diversity could have something to do either with higher plant diversity or with soil types. Most are soil dwellers, and some soil types are preferred for nesting. They're also likely to be specialists on some flowers. So please chime in, Katie.
That's a great point, and that may play a role in the high abundance of the solitaries at St. John's, because of course it's all fill.
Sometimes I think better when I write things down than when I say them. So
apologize for my phone.
Thank you for the question. Okay, any other questions for Susan? Okay, well, this has been a wonderful presentation. I have really enjoyed it. You just elucidated so much about how you pull this and everything else gets reorganized. It's just fascinating. So it's really I think it's going to be really helpful as we figure out how to try to restore some of these sites. So thank you very much, Dr. Watt.
Thanks so much for your attention. Thanks for your patience with my having to run in at the very last second.
Well, I personally am completely impressed that you were able to solve that problem just like that and be here with only 2 minutes after the hour. I'm stunned.
It was a lucky stroke. So please, by all means, do visit our website. We always are really interested to talk to other folks who are interested in flower insect interactions. So www.quamisheco.com.
Okay.
The website was listed on the blog post about tonight's presentation.
Oh, great.
Okay.
Thanks, Carolyn. And we have a couple of people urging you to warm up, and thank you so much for for braving the cold to give your presentation. We really appreciate it.
Not at all. It's really fun. It's always just so fun to talk with people who care about these things. So I'm so glad you guys are out there doing what you do.
Thank you for giving us some difficult concepts so very clearly and interpretively.
Thank you.
Beautiful talk.
Thank you. Well, I hope our paths will all cross again sometime soon. Thanks for your attention.
Out on the prairie.
Yeah.
Thanks, Susan.
Good night, everybody.
Good night.
Good night.
Night, y'all.
Cheerio.